and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper. This is of shift 1 and I will be discussing the part C questions. These are from your cell biology, molecular biology section, and this exam was conducted in December 2024. So coming to the first question. The helicase activity of E. coli DNA B was investigated using the following two substrates 1 and 2. So this is substrate 1 and this is substrate 2, that is your DNA. Followed by gel electrophoresis and autoradiography. So this is it, the autoradiogram. The result of these experiments are depicted below. So these are the components that they have added and tested in autoradiography using different substrate 1 and 2. The following statements are made and you have to see which all of these are correct statements. So before coming to the statements, I will tell you that DNA B, it is a helicase and it is involved in DNA replication and it requires ATP. This is what you have studied. Now substrate 1, if you see, it has got a 5' 3' end which is radio labeled DNA, and this complementary strand is unlabeled. Similarly in case of substrate 2. But substrate 1, if you see, it is totally unwound DNA. It is double stranded and unwound. But here also you can see it is partially unwound. That is why these open ends on both the sides. Okay, so these are the open ends, so it is partially unwound. Now see the lane, last lane in substrate 1, that is this one. If you don't add anything and there is no boiling, this is the size of the double stranded DNA that you are getting. Now if you don't add any of these components and you undergo only boiling treatment, then you get single stranded DNA at this position. Okay. Single stranded DNA upon boiling, why you get? Because boiling will result in the separation of the strands. Now helicase, it is involved in separation of the strands. Okay. Now you see this lane, first lane. You add single stranded DNA binding proteins, DNA B and ATP also, but you don't undergo boiling. It doesn't separate into single strand. It is found in double stranded unwound form itself. So even upon addition of DNA B and ATP, there is no unwinding of the DNA in case of substrate 1. Okay, fully wound DNA. Now take the case of substrate 2. As usual, this is the substrate 2 lanes from here to here, and this is the autoradiography result that they got. So if you see the last lane, there is no components being added, no boiling. This is the size of the double stranded or partially unwound DNA. This is the size. But here if you do boiling without adding these components, okay, you get a single stranded form. But here you see, here you are not adding DNA B, you are adding only single stranded DNA binding proteins and ATP. You get two bands. One band is the top band, it is more brighter than the lower band. And this shows that even if you don't add DNA B, single stranded DNA binding protein is sufficient to cause unwinding of this partially unwound DNA. Okay, but all of the DNA is not getting unwound. Only a small percentage of it is getting unwound. That is why the difference in band intensity. The majority of it is remaining in partially unwound condition, but a few percent of it has become unwound also fully because upon addition of single stranded DNA binding protein. Now take this lane 2. You are adding only two components, but ATP is not added. It remains partially unwound double-stranded form. So if you don't add ATP to DNA B, then it is not going to function. Now if you add all the three components and there is no boiling, you get a single-stranded form of DNA. So this states that DNA B will act only on a partially unwound DNA, not on a DNA which is fully wound and perfectly double-stranded. It is not going to act. It will act on a partially unwound DNA. Now read the statements. DNA B can unwind only a partially unwound DNA. I already told you as lane 1 in substrate 2 and lane 1 in substrate 1. I already discussed that. So that statement is correct. Then single-stranded DNA binding protein inhibits the unwinding activity of DNA B. So single-stranded DNA binding protein inhibits the unwinding activity of DNA B is not correct. Single-stranded DNA binding proteins, if you see this lane 3, this lane, you can see that only when single-stranded DNA binding protein are present but no DNA B, it is able to partially unwind the DNA. That is why you got a faint band over here. So it inhibits the unwinding activity of DNA B, you cannot say, because when you add single-stranded DNA binding protein and DNA B, you got a unwound DNA. Okay. So it doesn't inhibit the unwinding activity of DNA B. That is not correct. Then DNA B unwinds DNA in the 5' 3' direction. You cannot come to that conclusion from this experiment. Here if you see, this side is also open, this side is also open. So you cannot come to that conclusion based on this experiment. Then DNA B requires ATP for DNA binding. That is also correct based on this lane. The ATP is not added, DNA is not unwound. It is in the double-stranded form itself. Okay, next one. A lac lambda hybrid system is developed to study the lambda repressor protein. Okay, that is your CI, in which the lambda CI gene is under the control of E. coli lac promoter and operator. So this is your CI. It is under the control of lac promoter and operator. And the LAC-Z gene is under the control of, okay, the LAC-Z gene, which was usually under the control of the lac promoter and operator. They took it and put it under the control of lambda PRM promoter and OR operator. Now OR operator, there is operator 1, operator 2 and operator 3. Both the plasmids are introduced in E. coli. So you took one single E. coli cell, introduced both the plasmids into it. And the concentration of the protein are determined upon addition of IPTG. So this is the graph that you get after plotting the concentration of protein against IPTG. So this is the protein lambda repressor and this is the LAC-Z protein. Okay, now before coming to that question, I will tell you one thing. Now you all know the lambda repressor, that is CI, it exhibits both positive and negative feedback regulation of its own gene. Okay. Now what is this positive feedback regulation of the lambda repressor? So When C1, that is when the repressor concentration, C1, CI repressor is less, it will bind to operator OR1, OR2, activating transcription from the PRM promoter. So RNA polymerase will bind to the PRM promoter. It will result in the transcription translation of the CI gene. So the CI concentration increases. So and it will help to maintain the lysogenic state in the bacteriophage. Okay. Now at higher concentration, now PRM promoter has started synthesizing CI repressor gene and it has reached up to a certain level, and there is a higher concentration of CI in the cell. So what will happen is the CI, it will bind to operator O3. CI has got a higher affinity for operator 1 and a weaker affinity for operator 3. So when its concentration increases, there are more CI molecules available to bind to operator 3, and it will bind there, and this will interfere with the binding of the RNA polymerase to the PRM promoter, and thereby it will repress the transcription of CI. So CI is having negative feedback regulation of its own expression. Okay. So positive feedback means low concentration of CI, operator 1, operator 2 active, RNA polymerase bind, CI produced in more concentration. Once concentration increases, more CI molecules available, bind to OR3, and it will repress the transcription of CI. So whatever CI was present in the cell, it remains a constant. There is no increase and decrease upon binding of CI to the operator 3. Okay, so that is what is happening in normal condition. Now what is it with your IPTG? Now IPTG, it binds to the LAC repressor. Okay, so this is IPTG, it will bind to the LAC repressor protein which is bound to the operator. So upon IPTG binding, the LAC repressor will detach from the operator, and it will allow the RNA polymerase to bind to the promoter and initiate the transcription and translation of the structural genes. So here the structural gene is lambda CI. Okay. Now what will happen in your E. coli cell which is having both these plasmids in it? So you add IPTG, I told you it will result in the synthesis of lambda C1. So first there will be lesser concentration of C1 starting first. So what will happen? Okay, whatever less C1 was produced, it will go and bind to operator 1 and 2. More affinity, low concentration of C1. So PRM promoter is getting ready to bind with the RNA polymerase. It will bind and it will result in the synthesis of LAC-Z. Okay, LAC-Z concentration is increasing, CI concentration is increasing, LAC-Z concentration also increasing. Okay, now here this IPTG is resulting in the production of lambda CI. Now CI concentration increases in the cell. It is keeping on increasing. So more molecules of CI are available now. So CI also has a tendency, when concentration of CI is high, to bind to operator 3. It will bind there. Now upon binding to operator 3, it results in repression of synthesis of the gene LAC-Z. Okay. So whatever protein was produced, that is whatever LAC-Z protein was produced in the cell, it will remain constant. Okay. So what should be your graph here? So if you see this graph A, okay, in every case, okay, LAC-Z only has changed. the lambda repressor is always increasing. Okay, fine. So you have to scratch your head for only the LAC-Z version. Now what will happen to LAC-Z? Lambda repressor, it will keep on increasing. I told you lambda repressor increases at higher concentration. Slowly, LAC-Z production also is increasing as a result of lambda C1 CI repressor concentration increases. It keep on increasing. Then more molecules available to bind to the operator 3. They will bind and they will result in repression of the LAC-Z transcription. As a result, whatever LAC-Z was formed in the cell, it remains constant. There is no increase of LAC-Z after binding of the CI to the operator 3. So it will remain constant. This one is not going to be correct. There is no decrease in the LAC-Z concentration upon increase of the lambda C1 repressor. Here also it is not going to be negative. So this one also not correct. Here it is not going to keep on increasing. It will repress because operator 3 is also present. So your answer is option C. That is the graph. So 3 is the answer. Then next one. The following statements were made regarding the roles of histone modification in transcriptional regulation. Okay. Acetylation of histone is generally associated with transcriptional repression by making the chromatin more compact. Okay. Histone acetylation, it is associated with transcriptional activation and not repression. As a result of this acetylation, there is relaxation of the chromatin structure and it will make the DNA accessible to the transcriptional machinery. So A is not correct. You have to see for all the correct statements. Now methylation of histones can either activate, so that makes 1 minute, that makes 1 also wrong, 4 also wrong. Okay, now we have to see 2 and 3. Methylation of histones can activate or repress transcription depending on the specific residue modified. That is correct. Now if methylation is on lysine 4 of histone 3, then it is associated with transcriptional activation. Okay, now for the same histone 3, we are methylating lysine 9 and or lysine 27. Then it is associated with transcriptional repression. So methylation of histones can either activate or repress transcription depending on the specific residue modified. It is correct. Then phosphorylation of histones occur in response to DNA damage and can influence gene expression. So that is also correct. If you see the option 2 and 3, you can see there is B present, C also present. B present, C also present. So B and C are naturally going to be correct. You have to scratch your head only for D. Okay, now C is also correct. So when DNA damage occurs, there are certain kinases like ATM, ATR, etc. which phosphorylate specific histones. So this phosphorylation of histones makes the chromatin loose and it is made accessible to various DNA repair proteins. So that statement is also correct. Then histone modification influence the recruitment of RNA polymerase complex, okay, but not transcription factors. That is not correct. Because histone modification, it will influence both the recruitment of RNA polymerase as well as your transcription factors. So D is not correct. So B, C is the answer. D is also, third one is also not correct. Then next one. Okay, this is with regards to your attenuation. Okay, given below are different type of genetic manipulations of E. coli tryptophan operon. That is your column X. Okay. So column X is manipulating the E. coli tryptophan operon. So various manipulations they have done here. And there are consequences on its transcription. Okay, now what will happen to transcription is given in column Y under high tryptophan concentration. That is the highlight. They have not told regarding low tryptophan concentration. So these are taking place under high tryptophan concentration. So this is the tryptophan operon, okay, promoter, operator, leader sequence, attenuator 1, 2, 3, 4, E, D, C, B, okay. That is correct with regards to tryptophan operon. Figure has not changed. Which of the following options represent correct matches between column X and Y? Okay. Now before coming to the question, I will tell you what is happening at high level of tryptophan and low level of tryptophan. What is this attenuation? So attenuator, it is located in the leader sequence in mRNA and transcribed from the tryptophan operon. You all know that, and it has 4 regions, 1, 2, 3 and 4. And these 2 and 3 are free to form a hairpin loop, and 3 and 4 also can form hairpin loop. Now and this hairpin loop formation, it will vary depending on the high concentration of tryptophan and low concentration of tryptophan. Okay, now first we will see what is happening at low tryptophan concentration. So this leader peptide, it is having a tryptophan codon, that is over here. 2 TRP codons are there. Now the low tryptophan condition, this ribosome, it will encounter this TRP codons and it will stall there. The ribosome stalls there. It will stop there. Okay. So this prevent the region 2, that is this sequence 2, from forming or getting associated with the ribosome complex. It is free there. It is not associated with the ribosome complex. So it will have a tendency to form a hairpin loop structure with region 3. And it will form a hairpin loop structure, and this is called as the anti-termination hairpin loop. So because of this formation of this anti-termination hairpin loop, 3 and 4 cannot pair with each other, and this will allow RNA polymerase to bind, and TRP structural genes are transcribed, translated. That is what is happening in low level of tryptophan. Now high level of tryptophan, the bacteria is having sufficient tryptophan available. It is having abundant supply of charged tRNA carrying tryptophan amino acid. So what will happen here is there will be translation taking place in a faster translation will be there, because there is high level of tryptophan tRNA being available. So the sequence 2, it becomes associated with the ribosome complex, and it will not be available to form a hairpin loop with region 3. So region 3, what it will do, it will form a stem loop structure with region 4, and this stem loop structure is called the termination hairpin loop, and it will prevent the RNA polymerase from binding, and there will be no transcription and translation of the tryptophan structural genes, and attenuation result. So high level of tryptophan, 3 and 4 regions are getting paired, resulting in termination hairpin loop and attenuation take place. This is what is happening in high level and low level of tryptophan during attenuation. Now see the options. So inserting bases between leader peptide gene and sequence 2. So somewhere over here you insert a few bases. Now upon insertion of these few bases, what will happen is the 1 and 2 sequences, they will be separated away. That is, the spacing between them increases. Okay, spacing between them increases. Now as a result of that, the sequence 2 cannot get associated with the ribosome complex. So what it will do, it will free to pair with region 3. So 2 and 3 will pair together. It will not result in a termination loop, but it will result in an anti-termination loop. So attenuation will not take place, or I can say it is less attenuation. So A will be 2. Okay. So that makes option 4 itself as the answer, because A2 is not there in 1, 2 and 3 options. Okay. Now we will see what is happening in others also. Then the next B, inserting bases between sequence 2 and 3. So 2 and 3, here you insert a few bases. So as a result of this, what will happen? 2 and 3, they separate, and spacing between them increases, and they will not be able to form base pair with each other. So that makes 3 available to form base pair with 4. 3 and 4 base pairing occurs. Termination loop is formed. Attenuation happens. So B is more attenuation. So B will be 3. Now deleting sequence 4. Okay. So termination loop is not going to happen at all. There is no termination loop. 3 is not able to base pair with 4 because 4 itself is not there. How will it base pair and form the termination loop? So there is no attenuation. So C will be 4. No attenuation. So C4. Then elimination of the ribosome binding site for the leader peptide. So here if you there is a ribosome binding site that you eliminate, so it will result in complete attenuation only. Okay. So here what will happen? The ribosome will bind and it cannot translate the leader peptide. So it will result in complete attenuation. So D will be 1. So that makes option 4 as the answer. Okay, the next one is regards to your lac operon. You know there is a promoter, there is an operator. These are the structural genes. Now once the structural genes are transcribed, you can find that each structural gene has got an Shine-Dalgarno sequence upstream of the AUG codon. So Z is having a Shine-Dalgarno sequence upstream of its AUG codon, which is where the ribosome will bind, and it will result in the transcription translation of beta-galactosidase. Now Y is having a Shine-Dalgarno sequence here. Ribosomes bind, transcription translation of the Y gene, which result in the protein permease. Similarly, A is also having a Shine-Dalgarno sequence here. Ribosome binding site, transcription translation take place, and it result in the formation of trans-acetylase protein. So that is what is happening in case of lac operon. The rest of the story I am not telling with regard to lac operon. You all know it. So we will see the question now. This much you remember. Okay, the mRNA of the E. coli lac operon contain the open reading frames for lac Z, Y and A genes from a single cistron. You already saw that. It is observed that lac Z is translated more frequently. Okay, that is the highlight. It is translated more frequently than lac Y and A. That doesn't mean lac Y and A are not translated. They are also translated, but lac Z is translated more. So the concentration of the lac Z protein, that is your beta-galactosidase, is more than that of Y and A. Okay, or I can say the which of one of the following statement best describe the reason for this observation? So if you see the first statement, okay, we will see which is the correct statement. The Shine-Dalgarno sequence is present upstream of lac Z but not lac Y and A. That is not correct. I already told you lac Z has got its own Shine-Dalgarno sequence, Y has got its own Shine-Dalgarno sequence, and A is also having its own Shine-Dalgarno sequence. So that makes option 1 as the wrong answer. Then next one. Inhibitory RNA structures are present upstream of the AUG codon of LAC-Y and LAC-A, but not LAC-Z, affecting translation initiation rates. Now, if inhibitory RNA structures are present upstream of the AUG codon of LAC-Y and LAC-A, then they will not be translated at all. But here they have told in the question that LAC-Z is translated more frequently than Y and A. That is what they have told. They have not told that LAC-Y and A are not translated at all. They have told that Z is translated more. So 2 is also not correct. Then variation in the Shine-Dalgarno sequence upstream of LAC-Z, Y and A have different affinities for the ribozyme, affecting translation initiation rate. 100% correct. Now I told you each structural gene has got its own Shine-Dalgarno sequence, and suppose if there is a variation in the sequence of this Shine-Dalgarno sequence, which is present upstream of each Z, Y and A, the ribosome will have a tendency. The Shine-Dalgarno sequence variation is such that the ribosome has got more affinity towards the Shine-Dalgarno sequence which is present upstream of LAC-Z than LAC-Y and LAC-A. So the ribosome, it is having affinity to Shine-Dalgarno sequence present upstream of LAC-Z, and it will result in translation of the LAC-Z more frequently than LAC-Y and A. Whereas that means Shine-Dalgarno sequence present upstream of Y and A has got somewhat a lesser affinity for ribosomes. Okay, they bind there, but affinity when compared to that of LAC-Z is less. So it will result in different frequency of translation of the genes. So that is what it is meant by option 3, and that is your correct answer. Inhibitory RNA structures are present, again the same story, in Y and A coding sequence but not LAC-Z, affecting translation. So that is also not correct. Now, okay, next one. Cells have both reversible and non-reversible post-translational modifications. The following statements were made regarding the reversibility of post-translational modifications. Okay, there are various post-translational modifications like ubiquitination, ADP ribosylation, myristoylation, then prenylation, etc. And there are some others also. I am not going into detail of all the post-translational modifications, only those what have been asked I will be explaining. So ubiquitination, you know, it is it take place at the end terminal of methionine and lysine, and it is a reversible process, and the enzyme involved in this ubiquitination is ubiquitin ligase that adds ubiquitin molecules, and there is another one called as the deubiquitinase that removes the ubiquitin. So it is a reversible process. So this statement, ubiquitination of protein is reversible, it is correct. So that makes option C, that is ubiquitin of protein is irreversible, wrong. Okay, now see the other one you see, but ADP ribosylation of DNA is irreversible. Now ADP ribosylation, it occurs at glutamic acid and lysine. It is also a reversible process. So DP ribosyl transferase, it adds ADP ribose, and ADP ribosyl hydrolase removes the ADP ribose. So that makes option A wrong answer. So that means you can happily mark 1 and 2 wrong. So, okay, now we have to see B. Okay, I think answer is option 3. And sorry, option 4 will be the answer because C is wrong there. So 3 is also wrong. So option 4 will be the answer as per me. Okay, we will see. Now ubiquitination of protein is reversible, yes, and myristoylation of protein is irreversible. Okay, that is correct. Myristoylation, it involves the addition of myristate fatty acid to the N-terminal of glycine, and it is mediated by the enzyme N-myristoyl transferase. It is an irreversible process, so B is going to be correct. Then both ADP ribosylation and DNA prenylation of proteins are reversible. Okay, ADP ribosylation is reversible. But what about prenylation? Prenylation means lipid groups are attached to proteins, that is usually to the cysteine residues, and the enzyme involved is farnesyl transferase and geranyl transferase type 1. So, and it is an irreversible process. So that makes option D as the wrong answer. Then E, both prenylation and myristoylation of proteins are irreversible. That is correct. So that makes B and E as the answer. Option 4 is the answer.